Good morning, everyone. Today's topic is intercellular communication part 2. You know the objectives? So today we'll discuss about communication by chemical signals, ligand gated ion channels, G protein coupled receptors, catalytic receptors, nuclear receptors and second messengers. So the above slides we have seen in first class. Now, intercellular communication. Why intercellular communication is necessary? Unicellular organisms like amoeba function, performs all its functions like phagocytosis, digestion, nutrition, everything, reproduction, etc. by single cell. So it doesn't need to communicate with other amoeba for its living. But when it comes to higher organisms, coordination, different organs and organ systems have developed, and coordination between different organ systems is necessary for normal performance or normal living. For example, if a person feels like having food, the thought is generated in the brain and it commands the muscular system and the joints or skeletal system to go in search of food. Then he will take the food, put it in the mouth. For that, neural communication is occurring. From the nervous system, command is going to the skeletal system. So that is neural communication via synapse and neuromuscular junction. From one neuron to next neuron, one neuron to next neuron is via synapse, and neurons to the cell, muscle cell is neuromuscular junction. It is called neural communication. Then after the person keeps the food in his mouth, he starts chewing and he swallows. Voluntary action is also by neuromuscular junction, brings about voluntary function. Then second part of swallowing and third part of swallowing are involuntary functions. It involves autonomic nervous system. And here, if you take from upper esophagus to the lower esophagus, the food is transported via peristalsis. So peristalsis requires coordination of the smooth muscles of the gut, that is esophagus, smooth muscles. So they are very small smooth muscles, and peristaltic wave of contraction must start in the upper part of esophagus and end in the lower part of esophagus. For that, intrinsic nervous system acts, and the cell to cell communication is via gap junctions. Direct cell to cell communication between the smooth muscles occur. That is direct cell to cell communication via gap junctions. We have told about gap junctions previously. Then after the food enters the stomach and digestion occurs, if you take carbohydrate food, glucose content in the blood increases. So when the blood glucose level increases, it should be brought under control. So increased blood glucose causes secretion of hormone, it is called insulin, which will bring about, which acts on liver, muscle and target cells and glands. So adipose tissue to bring about the blood glucose level to normal range, within normal range. For that, hormones come under endocrine communication. So insulin secretion, when the insulin is secreted, it will act on liver cells to take up the glucose and convert glucose into glycogen, and the blood glucose level decreases. So that is endocrine communication. And lastly, the is insulin is getting secreted, and insulin secretion is also regulated by glucagon, and insulin inhibits glucagon secretion, glucagon stimulates insulin secretion. So these cells, glucagon cells, secreting cells, alpha cells, and pancreatic beta cells which secrete insulin are present in the islets of Langerhans, which are present close by. So there is no need for the blood to carry the or hormone or chemical substance from the site of production to the target cell. Here they are very close by. So the glucagon acts on beta cells to stimulate the insulin secretion. It is called paracrine secretion. Paracrine, nearby. Nearby cells, glucagon is released into ECF. In the ECF, it reaches the beta cell, stimulates the insulin secretion. That is, glucagon stimulates insulin secretion. But when the insulin secretion is increasing, after reaching a certain level, this insulin which is produced by the beta cell binds with insulin receptor on the beta cell and inhibits the beta cell from further secretion of insulin. It is called autocrine communication. Autocrine. It is similar to paracrine communication, but a special type. It is called autocrine communication. Auto, self, crine, secretion. So with this we'll start. With this introduction, we'll start intercellular communication. Now, cells communicate with each other via chemical messengers that include amines, amino acids, steroids, polypeptides, lipids, nucleotides, etc. Some messengers move from cell to cell, what I told you, gap junctions, without entering the extracellular fluid. And some messengers are secreted into the extracellular fluid and affect the functions of the neighboring cells. The intercellular communication mediated by messengers in the ECF are of three types. Neural communication, communication via synaptic junction. Paracrine communication. Here, local hormones diffuse in the ECF to affect the neighboring cells. Glucagon affecting the beta cell. Example. In addition, if the product of the cell binds to the receptors on the same cell, it is called autocrine communication. Example, insulin. Endocrine communication, in which hormones reach the target cells via the circulating blood. That is endocrine communication. In this picture, you can see direct cell-to-cell communication. The first one, the topmost, is the direct cell-to-cell communication, gap junctions. Second one is paracrine communication, nearby cells. The chemical agent mediator is acting on the nearby cells passing via extracellular fluid. The third picture, it is autocrine communication, where the product of the cell is acting on the cell. And coming to endocrine communication, here the endocrine gland secretes the hormones which enter the blood and reach the target tissues, target cells. Finally, he is showing synaptic transmission or neural communication where synaptic left, you can see presynaptic neuron communicating with postsynaptic neuron via synaptic left. Here, this from synaptic vesicles, the chemical mediator or neurotransmitter is being released. Mechanisms by which chemical messengers act. So now we know that chemical messengers are the signaling molecules which bring about physiological effects. But how these chemical messengers are identified? By the target cell. So target cell contains receptors which on binding with the chemical messenger brings about a physiological change. Now, that chemical messenger, external messenger, extracellular ligand are first messengers. Any molecule, ligand is any molecule which any signaling molecule is ligand. So, receptor ligand interaction is the beginning of the cell response. This event is transduced into four broad categories. How it will be, how this response occurs. It is via ion channel activation or opening or closing of ion channel, and G protein activation, then activation of enzyme activity within the cell, or direct activation of transcription. So transcription is production of mRNA from DNA, and this mRNA will bring out protein synthesis. Here protein synthesis can be structural proteins or enzymes, or structural proteins or enzymes which bring about change in the target cell. Okay. The extracellular ligands are called first messengers, and intercellular mediators are called second messengers. This is the first step of intercellular communication, that is ligand binding to receptor and receptor identifies the ligand and brings about response. So here the yellow one is the ligand, blue is the receptor. When ligand binds with receptor, it will cause changes in the uh, intercellular, either G protein or ion channel or catalytic enzyme or nuclear receptor, and it will cause response. Ligand gated ion channels. They open and close on ion binding with ligands. Sorry, they open and close on ion binding, ligand binding with the receptor. So, example, action of acetylcholine on motor end plate. I'll show this picture now. G protein coupled receptors, GPCRs. G protein or GTP binding protein are membrane bound nucleotide regulatory proteins. G proteins are proteins, globular proteins or heterotrimeric proteins having three subunits alpha, beta and gamma. So when alpha subunit binds with GDP, the three subunits associate together. The G protein gets activated when specific ligand binds with the receptor. Then alpha subunit exchanges GDP for GDP. So when alpha subunit binds with GTP, it releases GDP. It binds with GTP and unbinds with GDP, and when this happens it dissociates from beta and gamma subunits. When it associates with GTP, it dissociates with beta and gamma subunits. I have kept this picture in the middle, so let me show. extracellular fluid on one side, cytosol on the other side. You can see the cell membrane, bilapid cell membrane. In between, you can see two pictures of the ion channels. So first one, the messenger or ligand is not binding with the receptor. So the receptor and ion channel are the ion channel is closed because receptor is not binding with the or messenger is not binding with the receptor. Second diagram, the messenger now binds with the receptor. This receptor is ligand gated ion channel. So on binding with the messenger, there will be structural change, conformational change in the ion channel, and the ion channel opens, thus allowing the sodium, potassium chloride ions through it. Okay. Now we'll go back to G-protein coupled receptors. So here you can see G-protein coupled receptor. The violet one is G-protein coupled receptor. When it is associated with G protein, heterotrimeric G protein. So it is binding, alpha unit is binding with GDP. So alpha, beta and gamma are together. So in second picture, to the right, the G protein is coupled receptor is binding with signaling molecular ligand. So from G protein coupled receptor, GDP is going away and GDP is binding with the G protein. So here has not shown clearly, but when this happens, alpha subunit separates with beta and gamma subunits, and it will activated alpha subunit now activates inactive enzyme, intracellular enzyme, membrane bound intracellular enzyme. The third one, this activated GTP and alpha subunit G protein is now activating the enzyme. Inactive enzyme is being activated by the activated alpha subunit, and it will bring about cell response. So after the cell response, GTP breaks into GDP and inorganic phosphate. When this occurs, the alpha subunit goes away from the activated enzyme, making it inactive, and it associates with alpha, beta and gamma subunits when it binds with GDP. So, alpha subunit modulates the activity of target cell in three ways. So, the protein over there, inactive protein over there is can be adenylyl cyclase or phospholipase C or cyclic GMP, etc. So, it modulates alpha subunit modulates the activity of target cell in three ways. One is interaction with adenylyl cyclase or interaction with phospholipase C or directly opens an ion channel. Coming to the ligands, excuse me. Coming to the ligands for G protein coupled receptors, they are catecholamines, epinephrine, norepinephrine, that is adrenaline, noradrenaline, acetylcholine, TSH, and it can be light also. It can be light, etc. Coming to intercellular communication with catalytic receptor. So what is a catalytic receptor? It is an enzyme linked receptor and is a transmembrane receptor. That is, it will be in the membrane. It will cross the membrane. It will be on either side of the membrane within the membrane. It is a transmembrane receptor where the binding of an extracellular ligand causes enzymatic activity on the intracellular side. Extracellular ligand binds with the transmembrane receptor on the extracellular side, and intercellular side enzymatic activity occurs. Hence, a catalytic receptor is an integral membrane protein processing both enzymatic, catalytic and receptor functions. Coming to the second messengers, they generally activate protein kinases. These are enzymes that catalyze phosphorylation of tyrosine or serine and threonine residues in the proteins. These are amino acid residues in the proteins. Example, protein kinase C, protein kinase A, myosin light chain kinase. Second messengers are calcium ion, cyclic AMP, cyclic GMP, IP3, DAG, etc. IP3 is inositol triphosphate, DAG is diacyl glycerol, etc. These are second messengers. Coming to how this catalytic receptor is activated. You can see in the first picture, inactive catalytic domain on the intercellular aspect, and the outer cellular aspect there is ligand binding site. Ligand binding site, or you can say ligand binding site. So when it is a dimer, when the signaling molecules form a dimer, the two enzymes, they come close together, bind with the signaling molecule, and intracellular aspect of the signaling molecule, intercellular aspect of the catalytic domain becomes active. Intercellular part becomes active. Extracellular part binds with the ligand or the signaling molecule. Intercellular part becomes active. So, and brings about catalytic changes. Or it can also be like this: the signaling molecules bind with the receptor, and receptor is associated with another enzyme which gets activated on binding with the catalytic receptor binding with the signaling molecule. Now, nuclear receptors. These receptors are present in the nucleus or in the cytoplasm. After binding with the hormone or ligand, the receptor hormone complex or the ligand complex, receptor ligand complex, enters the nucleus and stimulate transcription of mRNA, and thus protein synthesis and exert physiological effects. Example of nuclear receptors or the hormones which act in this manner are steroid hormones and thyroid hormones. Even thyroid hormone is a steroid hormone. So they act in this manner. As the steroid hormones are fat soluble, they can enter the cell membrane and bind with the receptors and bring about change. In this picture, you can see growth factor. First, B, it is growth factor, and you can see the receptor over there. On binding with the growth receptor, growth factor binds with the receptor, and it brings about changes which are called MAP kinases. It will activate MAP kinase. It is written NAPK. It is mitogen activated protein kinase, MAPK, which binds with the transcription factor, or this, after certain changes, this transcription factor enters the nucleus, binds with the DNA, and brings about transcription of genes. that is B. C is steroid hormone entering the cytoplasm and binding with the um, receptor, forming receptor hormone complex which enters a nucleus, binds with the nuclear responsive element, and it will cause transcription of DNA and brings about physiological change. And D, you can see metabolite metabolite ligase. Those enter the cytoplasm, then enter the nucleus. This is nuclear receptor. Bind with the nuclear receptor, and heterodimeric nuclear receptor now causes nuclear responsive element to cause transcription of the gene, which brings about protein synthesis or enzymatic synthesis, which brings about the required physiological change effects, physiological effects. So this finishes the intercellular communication. If you have any doubts, you can write in the comment section. Thank you.